All right, Ninja Nerds, in this video, we are going to talk about the development of the heart. Before we go ahead and get started though, please continue to support us by hitting that like button, commenting down in the comment section, and please subscribe. Also, if you guys want to get in contact with us, down in the description box, we'll have links to our Facebook, Instagram, Patreon. If you guys want to support us, we truly appreciate it. All right, Ninja Nerds, let's get into it. All right, Ninja Nerds, so what we're going to do is we're going to talk about the development of the heart, but in order for us to do that, we're going to do this in kind of like sections. The first thing that we're going to try to do is to develop a heart tube, okay, one singular heart tube, and then surrounding that heart tube, we're going to try to develop what's called a pericardial cavity, okay? So that's the goal. So in order for us to do this, we have to start with this top of the diagram, and we're going to work our way down doing two different types of folding process. Don't worry, I'll explain it to you. The first thing I want you to look at here is the top of the embryo, right? When you're looking at the top or the superior view of the embryo, first thing I want you to remember here is you have two portions of this embryo. This portion here, very simply, is the cranial aspect of this developing embryo. And then this portion down here is the caudal aspect of the embryo. Another way of saying this is the head and this is the tail of the embryo. It's pretty simple, right? Well, if you guys remember, during the gastrulation process, remember cells from the epiblast are moving down through that primitive streak, right? We have the primitive streak right here and the primitive node right there. If you guys remember, some of these epiblasts, these blue cells, are moving through the primitive streak. And when they move through that primitive streak, do you remember what they do? They convert the hypoblast, if it's a bilaminar disc, it turns the hypoblast into the endoderm. And then it makes a new layer between this blue layer called the epiblast and the new endoderm layer, which is going to be called the mesoderm. That mesoderm is going to be sandwiched between the ectoderm and the endoderm. What I want you to understand out of all of this though, is that a collection of mesoderm really starts to kind of take its place in the cranial aspect of this developing embryo, right? So you're going to have kind of this really interesting mesoderm clumped here in the cranial aspect of the developing embryo in front of your prochordal plate. So technically what I want you to gain from this is the heart actually develops in the head and then it starts to move down into the thorax. We'll talk about that and you'll see that best with this type of view here with the sagittal view. But what I want us to do is to look at this actual development of the heart tube in two views. One is we're going to look here in the cross-sectional view. So we're going to kind of take like this. Okay, and we're going to look at it here in this cross-sectional view. Okay, and this is going to be very, very important. I really think this view is really, really important. The next thing that we're going to do is we're going to take and look at the actual development of the heart tube from a sagittal section. So we're going to take a sagittal section here. So now what we're going to be doing is taking a look at this in a sagittal section. Sagittal sections are good for being able to see the movement or the migration from the head into the thorax. All right, so we have two sections here that we're going to be kind of looking at to understand the development of the heart tube. Cross section and a sagittal section. Let's start here with the cross section. When you take this cross section, this one here, so we'll number it just so we understand. This is the first section, which we're going to be talking about here, and this is the second section, which we'll be talking about over here. When you take a cross section and you're looking at it after gastrulation has occurred, you have three layers here. This layer here is your ectoderm. This layer here in the middle is your mesoderm. And this layer here on the bottom is the endoderm. Or the blue is the ectoderm, the red is the mesoderm, and the green is the endoderm. When you look at it, you have particularly this mesoderm is the area that we really want to focus on. When you're looking at it, you have three components of it. This one here, which is next to this called the notochord, this is actually called your paraxial mesoderm. Okay? This one here, which is just lateral to it, is called your intermediate mesoderm. And this one over here, both of these sections, this one kind of coming up here and this one going down there, this is what's called your lateral mesoderm, your lateral plate mesoderm, if you will. To kind of give you guys an understanding here, this part of the lateral plate mesoderm is called your somatic. And then this one here is called your splanchnic layer of the lateral plate mesoderm. What I want you to understand is the heart tube develops from the splanchnic layer of the lateral plate mesoderm. So when we're talking about this mesoderm up there, really the mesoderm that I really want you to focus on, that is the cardiogenic area, is this splanchnic layer of the lateral plate mesoderm. Here's something very interesting that happens. This endoderm starts secreting lots of growth factors. So it starts secreting lots of growth factors that are going to try to influence this splanchnic layer of the lateral plate mesoderm. What are these growth factors that it's releasing? It's releasing a lot of what's called vascular endothelial growth factors. VEGF is another way that we kind of refer to it. And what VEGF does is it stimulates the lateral plate mesoderm to kind of start differentiating and specializing, if you will. What does that mean? Let me explain. Let's say imagine here that we're just taking the mesoderm, okay? So here's our mesoderm, okay? A clump of mesoderm. The vascular endothelial growth factor are going to do something very interesting where it's going to stimulate this mesoderm to kind of differentiate. And when it differentiates, it actually kind of does two things. One is it forms this outer core, and then one is it forms this kind of inner core. The outer portion is going to become what really will become our blood vessels and our heart tube, okay? And we call this outer portion here, we actually are going to call this the angioblast cells, okay? Your angioblasts. And then the inner part, which will become our blood cells, our red cells, white cells, all that stuff, this is technically what we call the hemocytoblast. So what VEGF is doing is it's causing the mesoderm to differentiate into what? Blood vessels, right? And actually the heart tube itself and blood cells. Now, let's actually take a look and see how VEGF actually influences the lateral plate mesoderm in the next step. So what we had here was the lateral plate mesoderm. VEGF was stimulating it, causing it to differentiate, make heart tubes. Now, take a look here. You see how from the lateral plate mesoderm, this actual chunk of mesoderm grows off of it. And it starts specializing, and you form little cavities. What are these cavities here? This cavities right here, these are called your heart tubes. These are called your heart tubes. And then these cavities here that are in the back, these are called your pericardial cavities. These are called your pericardial cavities. So from the lateral plate mesoderm, what did we do? We released VEGF, stimulated the lateral plate mesoderm to form heart tubes, and a little cavity starts to develop within that lateral plate mesoderm, and these are called your pericardial cavities. Now, what happens is this embryo, imagine these edges, I'm going to start trying to bring them closer to one another. So I'm going to bring these ectoderm layer, cause it to fuse together. Bring the mesoderm layer, cause it to fuse together. And bring these two edges here together. When I do that, guess what happens? These heart tubes are going to fuse together and make one singular heart tube. So two heart tubes come together and make one heart tube. These pericardial cavities, whenever these two edges come together, they fuse and takes two pericardial cavities and makes it into one pericardial cavity. And we'll just keep going here. Where did that actual green part? You know what this green part is actually? Ray said that's the endoderm, right? The endoderm will make the epithelial lining of the GI tract. That's going to get invaginated more posteriorly. And look, there's the actual endoderm lining, which will become the epithelial lining of our GI tract. So that's that step. Okay, so we took the lateral plate mesoderm, stimulated it with VEGF, formed heart tubes, pericardial cavities. During the lateral folding, the heart tubes, pericardial cavities fuse, become one heart tube, one pericardial cavity. One more thing that we have to understand. You see we have the heart tube kind of sitting in the pericardial cavity? Well, it doesn't just sit there without, it's going to just flop around. We can't let that happen. We have to kind of like hold it against this actual posterior wall a little bit. And so what happens is some mesoderm connects the pericardial cavity to the heart tube. What is this structure here called? This is called your dorsal mesocardium. So this is called your dorsal mesocardium. Okay, so we have our dorsal mesocardium, which is connecting the pericardial cavity to the heart tube. Let's now zoom in on the heart tube a little bit. So let's just kind of focus on the pericardial cavity and the heart tube. So again, here's the pericardial cavity, here's your dorsal mesocardium. When you look at the heart tube is actually made up of a couple layers. The most inner aspect of the heart tube, this red part, this is actually called the endocardium. It's made up of those endothelial cells which we developed from what? From the mesoderm, right? We said that the mesoderm gets stimulated by VEGF and makes angioblasts, and we also said hemocytoblasts. The angioblasts are what's going to become the endothelial lining and make the endocardium. On the outer part here, this blue layer here, this blue layer is called the myocardium. This is called your myocardium. And your myocardium is actually developed from cardiac myocytes. So this is cardiac myocytes. Now, what's very interesting is that these cardiac myoblasts, they start secreting kind of a little connective tissue or jelly-like substance which forms between the endocardium and the myocardium. What is that pink-like jelly substance which is made by the myocardium? That is called the cardiac jelly. Not even joking, it's called the cardiac jelly. Okay, so what we've done up to this point is we started with lateral plate mesoderm stimulated with VEGF, made two heart tubes, two pericardial cavities. From lateral folding, we fused the two heart tubes, made one heart tube, fused the pericardial cavities, made one pericardial cavity, suspended the heart tube in the pericardial cavity by the dorsal mesocardium, and if we zoomed in on the heart tube and looked at the layers, the inner layer is the endocardium, which is made from the angioblasts, and then you have the myocardium on the outside, which secrete a substance called cardiac jelly, which sits in the center. Boom, we hit this from the cross-sectional view. Now, let's take a look at this from the sagittal view. All right, so we took and looked at the development of the heart tube from what? In a cross-sectional view undergoing lateral folding. Now let's talk about the development of the heart tube in the pericardial cavity in a sagittal view as it undergoes cranial-caudal folding. So if we take a look here, again, we're looking at this section here. We're looking at a sagittal section. And again, we kind of numbered that 2. So first thing we have here is our sagittal section. Obviously up here in blue is your ectoderm. Here is your endoderm, right? Now, what I want you to remember is we're looking at this in front of, in the top, in the cranial-most portion. So here is that actual cardiogenic area or that clump of mesoderm which will become the heart tube. So right here is your clump of mesoderm that is going to become the actual heart tube. So this is basically your mesoderm which will become the heart tube. Now, what we're going to do is now that we understand that, there actually, one more thing here. You know how in between here, between this actual portion here, there's a little membrane, which is kind of connecting this cardiogenic mesoderm to the ecto and endoderm. You know what this little little stalk is here? That's called your buccopharyngeal membrane. That'll actually become the opening, right? The stomodium will become the mouth. Now, what happens here is that this mesoderm starts to undergo the differentiation process, right? How? Remember what the endoderm does? It releases VEGF. VEGF will then stimulate the mesoderm to do what? Start to form angioblasts, which would become kind of the endocardial lining, help to form pretty much your heart tube. And then inside of that heart tube, the other parts of the mesoderm will become your blood cells, your hemocytoblasts. So now go on to the next step. VEGF stimulated the actual mesoderm here to start differentiating. Now look what we have here. Still same thing here. We have our ectoderm, we have our endoderm. And again, remember, this is the cranial portion where the actual cardiogenic center is. This is the caudal portion. Now, the next thing you need to notice is, look what happened. It's no longer just one clump of mesoderm. Here on the top, you see how this is all dark? This dark portion here is what's going to become, that is the heart tube. And then this here, that's actually kind of like a little cavity. That is going to be the pericardial cavity. So it's the same process in the actual cross-sectional view, but you're just looking at this as it folds, something different happens, and I'll explain it. So, ectoderm, endoderm. Endoderm releases VEGF, stimulates the actual mesoderm to differentiate, forms a heart tube and a pericardial cavity. Here's where it gets really cool. The ectoderm and endoderm start folding. So they start folding at the ends, right? So the cranial end starts kind of turning in and the caudal end starts turning in. As it does that, it pulls this mesoderm structure towards the actual bottom part. It pulls it caudally. So then what happens is it moves from the head kind of a little bit more to like maybe the neck area. So now it's kind of towards the neck area. As it continues to keep folding, so now we have our embryo kind of folding here. Look where it actually goes. It completely turns in here and goes into your actual chest cavity. So during this process of what's called cranial-caudal folding, what is actually happening? You're actually moving. Imagine it like this. This is the best way I think to imagine it. Imagine your heart is developing above your head. As you start kind of folding, as the baby is actually folding, the actual heart is moving inwards in front of the face, in front of the neck, and into the thorax. That is what you're seeing in this sagittal section. Now, one more thing. Remember how we had the heart tubes fuse, pericardial cavities fuse? And basically the heart tube formed in the pericardial cavity. The same thing has to happen here. So what happens is here, you're going to have your pericardial cavity. During this cranial-caudal folding, guess what it does? It kind of pulls the actual heart tube into the pericardial cavity. So now here's my heart tube sitting in the pericardial cavity. Before, during this actual process, they were separated. But during the cranial-caudal folding, as it moves from the head, over the face, over the neck, and into the thorax, the heart tube gets stuffed into the pericardial cavity. So that is what I want you to understand. The last thing that we have to do here is what I want you to do is take the heart tube from this cross-sectional view, take the heart tube from the sagittal view, and let's yank it out and look at it in its lengthwise, what it actually looks like, and all the different kind of vesicular portions. So let's say that we take this heart tube here. We're just gonna, for right now, abbreviate them. We'll kind of denote them a little bit later and what they become, but I want you to kind of know this is the top of the heart tube, and then as we kind of go work our way down, this is the bottom of the heart tube. At the top of the heart tube, you have these little things that are coming off here. These are basically going to be your outflow tracts. So blood will actually come in from the bottom and then leave out via the top, okay? These outflow tracts are basically your dorsal aorta, okay? These are called your dorsal aorta. Okay, so same thing over here, I'm going to abbreviate it dorsal aorta, right? The dorsal aorta come from this area here at the top called the aortic sac. But really, this portion here at the top, most top portion here, we're going to abbreviate this TA. This is called your truncus arteriosus. So what I really want you to know is the truncus arteriosus really makes up the top of the heart tube. Below the truncus arteriosus is what's called your bulbus cordis. Okay, below the bulbus cordis is what's called your primitive ventricle. Below the primitive ventricle is the primitive atria. And below the primitive atria is what's called the sinus venosus. And then again, blood will come in via the sinus venosus, through the primitive atria, through the primitive ventricle, through the bulbus cordis, through the truncus arteriosus, and out through the dorsal aorta. So you kind of already start to get an idea of what some of these structures will become with respect to the adult heart. All right, so we've formed our heart tube, we've denoted the portions of it. Let's really dig into it a little bit more and talk about what these actual components become. All right, so we formed our heart tube. We've accomplished this. We know that the heart tube is coming from the lateral plate mesoderm, particularly the splanchnic portion of the lateral plate mesoderm. We know that it's under the influence of VEGF. We finally made it. Now, let's go back, let's denote these different portions, right? What did we kind of abbreviate them as? Just real quick here. Remember, this was the aortic sac, right? So the top part here is going to be what's called the aortic sac. And we said that the aortic sac goes into what? What structure does that actually fill into? We said that it actually leaves out via what structures? It'll leave via the dorsal aorta, right? Then below the aortic sac, what was the next structure? This was the truncus arteriosus. So then we have the truncus arteriosus. Here's what I really want you guys to know. The truncus arteriosus is what becomes the pulmonary artery and the aorta. So the truncus arteriosus is what becomes the pulmonary trunk, which is pretty much going to make up your pulmonary arteries, and your aortic arch. Okay? So, first thing I want you to know, truncus arteriosus becomes those two structures. What is below this? What do we say? The bulbus cordis. All right, so this is called our bulbus cordis. Now, what does the bulbus cordis become? This becomes the right ventricle, right? So this becomes the right ventricle and the right and left outflow tracts. So it's the actual outflow tracts from the right ventricle and the left ventricle. Very, very important. Okay, beautiful. The next structure here, below the bulbus cordis here in pink, is what? The primitive ventricle. So this is called your primitive ventricle. It's obvious what this is going to become. This was the right ventricle in the outflow tracts. What do you really have left ventricle-wise? This is going to become your left ventricle. The next thing below the primitive ventricle is going to be the primitive atria. It's obvious what these will become. The primitive atria is going to become both the left and right atrium. So it's going to become the left atrium and the right atrium. The last portion here, below the primitive atria, is called the sinus venosus. Before we kind of write that down though, the sinus venosus we said has inflow, right? So these are the inflow tracts. We'll name these in a little bit. But there's 3 inflow tracts entering into the sinus venosus via this horn and this horn. This is going to be the right horn of the sinus venosus, and this is going to be the left horn of the sinus venosus. But the names of the veins that are actually entering into it are the same. So again, what is this structure here called? It is called the sinus venosus. Now, coming in via these horns, what are the names of the veins that enter into it? You have the common cardinal veins. So you have what's called the common cardinal veins. And then generally, that's going to be the ones that are on the most outer part. Okay? Then if you come inwards, you have the umbilical veins. So then you'll have the umbilical veins. And then as you come the most inward, these will be your vitelline veins. These will be your vitelline veins. Beautiful. So now, do you remember how I told you that once you kind of have an understanding at this point, it's pretty easy that blood is coming in via these veins into the sinus venosus, which will empty into the primitive atria, go to the primitive ventricle, go to the bulbus cordis, which is pretty much going to make your outflow tracts. That'll then spit that out into the pulmonary trunk, into the aortic arch, and then eventually you'll have what's called the aortic sac and the dorsal aorta, right? Now, does this look like what a heart looks like? No. So now what we have to do is there's a process called cardiac looping. So now what we have to do is go through a process called cardiac looping, which is annoying as all crap. But what I really need you guys to take away from this is the reason why cardiac looping is important is it depends upon particular types of proteins called dyneins. Okay? And if these dyneins are absent, it can lead to what's called cardiogenic syndrome. And this is basically where someone can have, they either have absent cilia, but the big part here is that the heart doesn't actually loop properly and form properly. And you know what happens? It actually doesn't normally, for a normal person, the heart will kind of actually bend kind of 2/3 to the left of the mid-sternal line. If you have some type of issue with these dyneins, guess what happens? The heart doesn't actually bend to the left, it bends to the right. And this can lead to dextrocardia, or sometimes what's called situs inversus. Okay, so it's important to understand why cardiac folding is important, because you need these dyneins to allow for that to occur. All right, nonetheless, what I want you to take into consideration here is we're going to start this cardiac looping process. Let's kind of denote these. Just the truncus arteriosus here. Right, truncus arteriosus, this is our bulbus cordis, this is our primitive ventricle, this is our primitive atria, and then here at the bottom you'll have your sinus venosus, okay? What I want you to know here is that what happens is that the truncus arteriosus and the bulbus cordis start kind of like moving to the right and downward. So the first part of cardiac looping is the truncus arteriosus and the bulbus cordis kind of start moving downward and to the right. All right, so truncus arteriosus and bulbus cordis move down and to the right. And by doing that, you start to get this configuration, okay? Then what happens is the bulbus cordis continues to kind of move downward. The truncus arteriosus continues to kind of move downwards and to the right. But then the primitive ventricle starts to move towards the left of the midline. So now the primitive ventricle is going to start moving to the left of the midline. So now look what happens. As that happens, the truncus arteriosus, bulbus cordis start moving a little bit towards downwards and to the right. So you get this kind of configuration here. The primitive ventricle is going to move left of the midline, so then this is going to be your primitive ventricle. And the primitive atria actually kind of get sucked into the back. They start kind of moving backwards. So then back here, you're going to have your primitive atria and your sinus venosus with your inflow tracts, right? Now, here's what I want you to understand, because it's very difficult to imagine this. You're looking at this from kind of a frontal view, okay? So you're looking at this kind of head on. If I were to take this and turn it where you were looking at this from a side view, you would get kind of something like this. So remember, what connects to the primitive atria above? The primitive ventricle. So here, let's have kind of the primitive ventricle here. And what happens is, here we'll have bulbus cordis, primitive ventricle, and then coming here backwards is going to be your primitive atria. And then coming off of that is going to be your sinus venosus, right? So what you actually see here is when you're looking at this view, the primitive atria is actually going to start moving backwards and then upwards. So the primitive atria will move backwards and upwards. And then look what happens here. All right, so we understand, right, with the primitive atria, it has to move where? It moves backwards and then it starts moving upwards, right? So as it starts to move backwards and upwards, they start to pop up over the bulbus cordis and the primitive ventricle. So now you have the primitive atria here, right? You still have your truncus arteriosus. You have your bulbus cordis. You have your primitive ventricle here. Right? And then again, you can't see it, but again, what will be coming off of the primitive atria coming behind this? This is going to be the sinus venosus, right? So it's starting to somewhat look like a heart, right? Because the primitive ventricle become the left, the primitive atria will form your right and left atrium, your bulbus cordis will form the right ventricle, and then the outflow tracts and the truncus arteriosus will make the pulmonary trunk and the aortic arch, right? So we're starting to get some of this view. Now what happens is, you got to remember, as this cardiac looping is occurring, it's actually kind of occurring inside of this pericardial cavity here. So what I want you to think about here is we have this type of pericardium here, right? This is our pericardium, okay? And you have the pericardium, you have the different portions of it, right? Like you have the outer fibrous pericardium, and then you have the inner, what's called parietal pericardium. Don't worry about that right now. What I do want you to understand is that if you guys remember, you had that structure here called the sinus venosus, right? The sinus venosus does two very interesting things. It starts to actually allow some of the cells within this area to move into the pericardial cavity. And as it moves into the pericardial cavity, it starts to form around the heart. Okay? And as it starts to form around the heart, you start to get this layer underneath of the pericardium. What do you think this is called? That's called your visceral pericardium. So the sinus venosus is giving way to what structure? The visceral pericardium. All right. One more thing that I also think is important. In the beginning weeks, like week 4, week 6, some of the cells of the sinus venosus even infiltrate into the heart and start to form some small, like, primitive conduction system that allows for the heart to start beating. And you can detect that on transvaginal ultrasound around week 6. So not only does the sinus venosus give way to the actual visceral pericardium, but it also gives way to what's called a primitive kind of conduction system that can be detected during the gestational development. All right, cool. Now, we've taken and developed our heart tube. We looped the crap out of it. We formed it inside of the pericardial cavity. We layered it with a visceral pericardium, and now it's starting to even have a little bit of a primitive beating type of activity. Now what I really want us to do is to take these primitive atria, right? And then your primitive ventricle and your bulbus cordis, and really start developing septations, or forming chambers that separate the atria from one another and separate the atria from the ventricles. That's the next step. Let's do it. All right, so now let's go ahead and take and start kind of first forming the next goal. We formed the heart tube. We looped it. We put it in the pericardial cavity. We made some layerings. We have some conduction. The next goal that I want us to accomplish here is to actually form what's called an AV, 2 AV canals. In other words, a canal between the atria and the ventricles, both a right AV canal and a left AV canal. That's the first goal. Let's do it. So if you guys remember, what was the structure here? You guys will know this by the end, right? So truncus arteriosus, bulbus cordis, primitive ventricle, primitive atria, sinus venosus. There's actually a little sulcus between the primitive atria and the primitive ventricle. So right here, I'm coming through that little sulcus there. That's called your AV sulcus. I'm making a section right here between the primitive atria and the primitive ventricle. What I want you to imagine is when I make this cut, this is what we're looking at. We're literally looking at the inner aspect of the heart tube, okay? This is going to be the posterior portion of the heart tube, and this is going to be the anterior portion of the heart tube. Now, the next thing I need you to imagine, out here, coming at you, like it's coming out into actually hit you in the face, is going to be the primitive atria, okay? Then going into the board is the primitive ventricle, okay? So you're looking at the posterior anterior portion in this view, coming out primitive atria, going in primitive ventricle. What I need you to first understand is that you guys remember those pesky little neural crest cells? I know you guys do. Those pesky little neural crest cells start actually kind of migrating here. So here, we're gonna draw a couple neural crest cells. So here's some neural crest cells. Those neural crest cells will start migrating into this area and start forming little kind of like cushions that come off the posterior portion and that come off of the anterior portion. So these neural crest cells are going to move in here and they're going to start forming like little cushions. Okay, what are these things here called? They're called endocardial cushions. These would be your anterior endocardial cushions, and this would be your posterior endocardial cushions, right? So here we'll put here endocardial cushions, endocardial cushion. This would be the posterior, that would be the anterior. As these endocardial cushions coming from the neural crest cells continue to grow, guess what they do? They fuse together. And when they fuse together, something very interesting happens, where you start to develop two little canals, right? So now I have a little kind of like canal here. A little canal here and a little canal here. And what was above here? Primitive atria. What was going into the board? Primitive ventricle. That's very interesting. We'll talk about what that is in just a second, but what I do need you to know is that when those endocardial cushions come together and fuse, they make a special name. We got to give it a special name. This is called the septum intermedium. So the septum intermedium is the structure that is formed when the posterior and anterior endocardial cushions from the neural crest cells come together and fuse. When they fuse, they now beautifully separate the primitive atria and the primitive ventricle and form two little canals. What are those little canals? Okay, let's do it again. Here's our septum intermedium, formed by those endocardial cushions coming together. And then here, on one side of the septum intermedium, is going to be an AV canal. This one here is called your right AV canal. And this one here is called your left AV canal. So now I have a separation of some form between the left side of the heart in some ways and the right side of the heart. Isn't that cool? Let's take a look at this in a different view. So now what I want you to do is I want you to take the heart like we had over here and kind of cut it, okay? Cut it like in a coronal section. And you're going to get this view here. So if we kind of take a look here, imagine this wasn't here for a second. Imagine that wasn't there. Up here would be your primitive atria, and let's just say here's primitive ventricle, here's part of that bulbus cordis again, right? So if you wanted to say this here, let's just say that this was part of the bulbus cordis. Let's say this was the primitive ventricle, and this was a primitive atria, this was a primitive atria. Remember what was forming between these two? But it was coming like this. What was it? Those endocardial cushions come together, and they made what? Septum intermedium. Now, here's what's really cool. Coming off of the septum intermedium, some of these actual endocardial cells start kind of forming these little like valves that come off of them. So they form like these little valves, and then they connect the valves together. You know how they connect the valves together? They make this thing called an annulus ring. And then from that annulus ring comes these little valve flaps. And then guess what comes from the valve flaps? Guess what comes off of those little pesky valve flaps? Your chordae tendineae. So now, from that septum intermedium, what did I do? I formed valves on both sides of it, and a valvular annulus and little chordae tendineae. I just formed the valvular apparatus. But I formed a valvular apparatus on one side, particularly this is the left side. Guess what this is going to become? Guess what this valvular apparatus will become? This will become the mitral valve. This will become the mitral valve apparatus, or sometimes referred to as the bicuspid valve. Guess what this one will become? The tricuspid valve. Oh man, isn't that cool? That is so cool. Okay, so now I formed the tricuspid valve apparatus. So from the septum intermedium, I formed a right AV canal. What is this canal here? Right AV canal. And over here I form a left AV canal. And off of the septum intermedium, what comes off? The mitral valve on the left side, tricuspid valve on the right side. And then also what forms along with those valves? The valvular apparatus, which is the annular ring, and the chordae tendineae. Oh, let's keep going. We've now formed a right kind of inflow from the atria to the ventricles, right? And we formed a valvular apparatus to provide, what is the purpose of the valves? To provide a one-way flow. In other words, to prevent backflow from these ventricles, in this case primitive ventricle or bulbus cordis, back into the primitive atria. That's the whole purpose. Now, the next thing that we have to do, we formed these kind of AV canals on the right and left side. Now let's separate the primitive atria. Let's form a right atrium and left atrium. Now this is I can't stress enough, this is so high yield. If there's anything that you guys take away from this, this is one of the big ones, because a lot of like diseases can develop because of a malfunction in this process. Okay, so from this point here we have our primitive atria. Let's actually kind of annotate this. Here's primitive atria on this side, primitive atria on this side, and then you still know you have bulbus cordis and primitive ventricle. Coming down the middle of this primitive atria, you get a little septum. And this septum almost comes all the way down to meet with this, what is this little brown structure here in the middle? That is our septum intermedium. So you have this kind of like tissue here that tries to grow downwards from the top of the primitive atria down towards the septum intermedium, but doesn't actually reach it. What is this tissue here called? This tissue is called the septum primum. It's called the septum primum. And because it doesn't reach all the way down to the septum intermedium, it creates a little space, if you will. What is that little hole there called? Well, guess what the name of this was? Septum primum. You know what the name for a hole is? Ostium. So it's ostium primum. So this little hole here is called the ostium primum. Beautiful. Okay, so in this first step of developing chambers between the atria, you have a septum primum come down to approach the septum intermedium, doesn't reach it, creates a space called the ostium primum. What's the next thing that happens? Let's come up. We have again septum primum, but guess what? Septum primum continues to grow, and eventually it does reach the septum intermedium. And the septum intermedium was here in the center, and again, what color was it in this situation? It was brown. So what happens here? The septum primum finally went down, closed off the ostium primum. But guess what? Another hole develops, but it develops towards the top of the septum primum. What is this little hole here called? So this is still, what is this structure here called? We're going to abbreviate it septum primum. But a little hole in the septum primum develops towards the top after it completely moves all the way down. What is this little hole here called? This is called the ostium secundum. Man, crazy names. So this is called the ostium secundum. All right, so now we formed the ostium secundum within the septum primum. What's the next thing that happens? All right, let's get into it, right? Sounds like I'm naming transformers right now. Ostium secundum and the septum primum, huh? All right, let's get back into it. So first thing that we have here is, what do? We formed the... Dang you, Robert! Dang you! All right, so now, let's take your ostium secundum, huh? From the septum primum here. So we're going to put that down like there. So now we have our septum primum with this little space there called the ostium secundum. Right? So again, right here is septum primum. And then what is this little space here called? This is called the ostium secundum. Now, another thing happens here. Another piece of tissue starts growing. And this son of a gun starts coming down to try to block the ostium secundum. What is this one? It's another transformer. It's called the septum secundum. All right. So we got the septum secundum. What is this one here called? This is called your septum secundum. So now here's what's really important. We've now formed a septum secundum to block the ostium secundum. But there's still a little space here. You see this tiny little space like right here between the septum secundum and the ostium secundum. There's a name for that little structure there. This is called the foramen ovale. You know why that's really important? Is the baby's sitting in utero and amniotic fluid. It can't really breathe. So what happens is the blood will come into this, eventually the right atrium, bypass the right atrium, because you know the right atrium goes to the right ventricle, pulmonary circulation goes to the lungs. We don't need blood to go to the lungs when the baby's in utero that much. So what we do is we bypass the blood from the right atrium into what's called, eventually going to be the left atrium. Left atrium, left ventricle goes to the systemic circulation, which doesn't go to the heart. Go to the lungs, okay? Why is this important? Because eventually the foramen ovale will close. But if it doesn't, it can lead to what's called a patent foramen ovale. You know why that's important? Is because sometimes if people develop like little clots on their legs called DVTs, they can come up via the right atrium, go to the left atrium, left ventricle, and guess where they can go? Up into your brain and cause a stroke. It's called a paradoxical embolus. They're not very common, but again, it's if this does not close. Alrighty. So now we've developed a septation between the primitive atria. So now I can technically call this side what? The right atrium, and I can now technically call this side the left atrium. Oh, that's so cool. Now, let's separate our bulbus cordis and our primitive ventricle. Because we know primitive ventricle is going to become what? Left ventricle. Bulbus cordis should become the right ventricle. And then the other part, the superior part of the bulbus cordis becomes the outflow tracts. Watch this. All right, so now let's erase this. And again, what was this little tissue here? This was called your septum intermedium. At the apex of the heart, at the apex, you start to develop some tissue here that starts moving its way upwards from the apex. And this tissue here, as it moves upwards towards the apex, is given a particular name. We're trying to form another septum. But this septum that we're actually making here is called the muscular portion of the interventricular septum. What is this one here called? It's called the muscular portion, we're going to abbreviate this, of interventricular septum. Then, guess what? We still have a little space here between the bulbus cordis, which will become the right ventricle, and the primitive ventricle, which will become the left ventricle. This space has to get filled. So what happens is, from the septum intermedium, some tissue moves downwards and starts to fuse with this muscular portion of the interventricular septum. What is this tissue here called? This is called the membranous portion of the interventricular septum. Why is that important? Guess what? If the membranous portion of the interventricular septum doesn't come down and meet the muscular portion of the interventricular septum, guess what you develop? A little hole, a ventricular septal defect. So that's another reason why this is very important. So what have we done now? Technically, I now have this portion of the bulbus cordis is technically now the right ventricle. And now this primitive ventricle is technically now the left ventricle. Now remember, I don't want you guys to get confused. The bulbus cordis makes two things, right ventricle and the outflow tract. So we still have to talk about the other portion of the bulbus cordis. But one portion of it just made our right ventricle. What have we done? We took a heart tube, septated it to form an interatrial septum, an interventricular septum, a right AV canal, a left AV canal. Well, what do we got to do now? Let's get some inflow tracts coming into that right atrium. All right, so the next thing I want to do is I want to kind of develop an inflow tract into particularly the right atrium. So the next thing we have to do is take another section here. Remember, this was our primitive atria here. This was our sinus venosus. I'm going to come right around this level here, and we're going to look at this structure here. So I'm cutting it off here. You're just getting the top portion here of the primitive atria, or the bottom portion of the primitive atria, and then here you're still getting a little bit of that sinus venosus. Now remember, what were the veins? I'm not going to actually write them down. I want to see if you guys remember them. Coming into the sinus venosus, we had the inflow tracts. From the outsides, what were these? Your common cardinal veins. Here, what were these? Umbilical veins. Here, what were these? Vitelline veins. The common cardinals, the umbilical and the vitelline veins empty into their horns. This was the left horn, and this is the right horn. All right, so what I need you guys to understand is, again, we have the left horn, right horn feeding into the sinus venosus, which will feed into the primitive atria, okay? Now, remember, left horn, right horn are fed by three vessels, right? We had, again, what were these? Common cardinal veins on the outside. In the middle is your umbilical, and on the most inner part are your vitelline veins. What happens is all of the veins on the left horn break down. All that's left over is just the left horn. So now I just have the left horn. Okay? Then on the right horn, we're just going to abbreviate this RH. What happens is the umbilical vein actually starts to degenerate. And all that's left over feeding into the right horn, we'll abbreviate this as the common cardinal vein and the vitelline vein. So all that's feeding into the right horn is the common cardinal vein and the vitelline vein, and all the tributaries off of the left horn break down. The next thing is, not only do those actual tributaries break down, but the left horn starts kind of shifting towards the right and actually kind of fuses with the right horn a little bit before it enters into the sinus venosus. So again, here we had above this, where these horns enter in, your sinus venosus, and then above this would be the primitive atria. Now, here's what I want you guys to remember. When this is actually fusing right? Whenever these structures are all kind of like changing and going through their kind of digression and specializing, this sinus venosus gets absorbed into the primitive atria. So the sinus venosus will get absorbed into the primitive atria. So whenever the sinus venosus gets absorbed into the primitive atria, the left horn is going to develop a very special structure. Guess what this little structure is? This is called your coronary sinus. So the coronary sinus is formed from what? From the left horn. Then, again, whenever the sinus venosus gets absorbed into the primitive atria, the common cardinal vein is going to be up here towards the top. This common cardinal vein will be what's called the superior vena cava. The superior vena cava is formed from the right common cardinal vein. And then this bottom part here, remember how from the right horn you had the vitelline vein. Whenever the sinus venosus is absorbed into the primitive atria, this right here is the inferior vena cava. And the inferior vena cava is formed from the vitelline vein. Which one? Particularly the right vitelline vein. Okay? So that's what I wanted you to understand. So the inflow tracts coming into the atria, because of the left horn shifting towards the right and all of its tributaries breaking down and kind of feeding into the right horn a little bit before it enters into the sinus venosus, whenever that gets absorbed into the primitive atria, it gets absorbed into the right side of the primitive atria. And again, coronary sinus is formed by the left horn. The superior vena cava is formed from the right common cardinal vein. And then the inferior vena cava is formed from the right vitelline vein. Beautiful. And then these are taking blood and emptying them into the right atrium. Isn't that cool? So now not only have we formed left atrium, right ventricle, left ventricle, right AV canal, left AV canal, we also have the inflow tracts coming into the right atrium. Isn't that cool? Now we got to come to the annoying part. Now we finally have to go over the bulbus, cordis, and the truncus arteriosus and how they make those outflow tracts. Let's do that now. All right, so we're looking at another kind of, again, kind of a coronal section of the heart. And again, so you can kind of have an idea here. We've already formed our right atrium. We've already formed our left atrium. We have, let's say that we haven't covered, let's just kind of rewind a little bit and say at this point we haven't formed the membranous part of the interventricular septum. But for the most part, we have our right ventricle, we have our left ventricle here. And here you can see like these little canals. You see those little canals, those little holes there? That's your right AV canal and that's your left AV canal, okay? Now remember, we said that this portion of the right ventricle is made by the bulbus cordis, right? But we also said up here we have another portion of the bulbus cordis, and up here we have a portion of the truncus arteriosus, okay? What I want us to do is I want us to take a cut from here and here and really kind of look and see what is actually happening, that we form a defined aorta and a defined pulmonary trunk, and then on top of that, the valves that are going to be allowing for a one-way flow from the right ventricular outflow tract and left ventricular outflow tract into the respective pulmonary trunk and aorta. Okay, so if we take a section here and here, we get this kind of structure here. So again, at the top portion here is going to be your truncus arteriosus, and towards this bottom portion here is going to be the bulbus cordis. Okay? All right, beautiful. Now, what happens is you start to form little, again, neural crest cells. What are they called again? Neural crest cells start to infiltrate into these areas here. So some of these neural crest cells are important for forming what's called the aortico-pulmonary septum, but some of these neural crest cells, they kind of migrate into this area here, okay? When they migrate into this area, they start forming little like endocardial ridges or kind of cushions kind of coming outwards. At the truncus arteriosus, the top part, you get a truncal ridge that forms like this, okay? And then another truncal ridge that forms like this. Okay? At the bottom part of the bulbus cordis, you get a bulbar ridge that kind of comes in like this. And again, you get another bulbar ridge that comes in like this. Okay? Now, there's another ridge in here that has actually formed, but it's not in a view that we can see. This is forming like on a left side and a right side, kind of coming towards each other. There's another one in the middle. You can't see the way it's forming, but it's forming from like an anterior-posterior view. So what I'm going to do here is I'm just going to kind of draw it like this. So you have a ridge coming from the anterior portion, and you have a ridge coming from the posterior portion, and they're trying to approach one another. Whenever these ridges kind of approach one another and come together, they do this kind of like spiraling around the central axis. All right, so now what I want you to remember is that those truncal ridges, right, they come together. And whenever those truncal ridges come together at the top part, right, where the truncus arteriosus is, they kind of form this like little septum that separates this kind of whole, what is going to become the aorta and the pulmonary trunk, into two sections, which will again give us one portion of that little septum will flow into the pulmonary trunk, one of that section will flow into the aortic arch. But again, what happens here at this level of the truncus arteriosus here? Okay, this is going to be forming what's called truncal ridges. And again, what happens is the truncal ridges come together, fuse, and then kind of form a nice little septum there. Same thing at the bottom, right? Here at that bottom portion here where the bulbus cordis is. Again, you get those bulbar ridges coming from both of these edges, and they meet in the middle and form a nice little septum. Okay, so again, you have those nice bulbar ridges, they come together and they make a little septum. And then we said here in the center, still part of the bulbus cordis, technically another area we call the conus cordis. We're not going to get too much into that, but they form anterior to posterior. So we're just going to kind of draw a central line there. What I want you to now do is that because the way the conus cordis ridges form and lead to a septum, it causes this entire septum that runs down the length of the top from the truncus arteriosus to the bulbus cordis to kind of corkscrew. And so now follow this edge and have it kind of move through the middle here and kind of come to the other edge here. Come from this edge here, have that go through the center and meet at that point. Now, all this does is this creates a nice little septum here. What is this septum here called? This septum here is called the aorta. Let's actually erase this part here. This is called the aortico-pulmonary septum. So what is this called here? This is called the aortico-pulmonary septum. And it's basically just kind of separating the aorta from the pulmonary trunk. Pretty straightforward, right? Now, from the bulbus cordis and the primitive ventricle, we already said that the blood was kind of moving up through this, right? As it moves up through this, here's what's really interesting. The blood flow, okay, from the left ventricle will move through this posterior portion, okay, from this bulbar septum. It'll move via this posterior portion. When it moves via the posterior portion, it'll move through the back part of this aortico-pulmonary septum, so it'll kind of move under it. When it moves under it, it comes out from here like this, so it kind of moved under it from the back. When it does that, it then moves over the top part of this aortico-pulmonary septum and then out through this anterior portion. Okay, out through this anterior portion formed by that truncal septum. So this blood flow coming from the left ventricle, moving outwards and coming up through this front portion here, this is going to be giving way to the aortic arch. Okay? In the same way, let's follow this front portion here. So in the anterior portion of the bulbar septum at this point, the blood will be coming through this anterior portion here. When it moves through the anterior portion, it moves in front of the bottom part of the aortico-pulmonary septum. And then when it gets to this kind of like corkscrew point at the conus, it moves behind this top part of the aortico-pulmonary septum. And when it moves behind, it comes up through the back part of that actual truncal septum. And this is going to give way to the pulmonary trunk. And this blood is going to be coming from the right ventricle. So, the aortico-pulmonary septum is formed from neural crest cells that lead to little ridges, truncal ridges at the top, bulbar ridges in the bottom, forming from kind of like the left and right towards the center. And the center, what's called the conus cordis, there's ridges that are forming from posterior to anterior. Because the way these ridges actually form, and the center, anterior to posterior, and at the top and bottom from left to right, it creates a corkscrew type of fashion here, which is called the aortico-pulmonary septum. Blood flowing in through the back part at the bulbar region, where that septum is, comes underneath the bottom part of the aortico-pulmonary septum, and over the top of the top portion of the aortico-pulmonary septum out via the aortic arch. And then blood flowing through the anterior portion or ventral portion at the bottom part of the bulbus cordis will move over this bottom portion of the aortico-pulmonary septum and move under this top portion of the aortico-pulmonary septum and come out via the pulmonary trunk. The next thing that happens is this actual whole kind of aortico-pulmonary trunk-like structure will rotate. And so once it undergoes this rotation, it kind of splits these structures up. It splits them up. And then you get this beautiful little setup here. And this is how I remember this kind of blood flow, where it's moving with respect to this. Blood flow coming from the left ventricle moves up via the aorta. And where does it go with respect to the pulmonary trunk? It moves underneath the pulmonary trunk and then moves over the pulmonary arteries. So you can kind of follow that same flow with respect to the aortico-pulmonary septum. It goes underneath and then over. The same concept here with the pulmonary trunk. Blood is coming from the right ventricle, moving up via this pulmonary trunk. Where is it going with respect to the aorta? It's going above the aorta and then underneath the aorta. Look over here, it's going above and then going behind. So that's a nice little way to remember kind of that blood flow pattern. Now, let's take this structure now. We formed our aorta, we formed our pulmonary trunk, we start having our outflow tracts. Let's now just connect that to the ventricles. And you now see this beautiful little setup here. Look what we've made. We've made a right atrium. We've made a left atrium. We've made a left ventricle, a right ventricle. The right ventricle formed from the bulbus cordis, and again, these top portions here, this portion here and this portion here. The outflow tracts from the right ventricle and the outflow tracts from the left ventricle are made via the bulbus cordis. So this is your outflow tract, then coming out into the pulmonary artery, coming out here into the aorta. Beautiful. Look at that. We've made something here. It's pretty cool, but we're not done yet. Now what we have to do is we have to form little valves between the ventricles and their corresponding arteries. Let's do that now. All right, Ninja Nerds, we are so close to being done. Okay, the next thing we got to do is we got to start talking about making the semilunar valves. So where we got to go is, again, go back to this structure here where the truncus arteriosus and the bulbus cordis is. We're going to go right here, kind of along that junction. Okay, technically, remember I told you there's the bulbus cordis, and then technically in the center between the bulbus cordis and the truncus arteriosus is called the conus cordis. We're going to make a section right there. So what we're looking at here is a cross section at the bulbus cordis and conus cordis junction. It's one heck of a thing there. Okay, all right, so what we're looking at this in view-wise is this anterior here, posterior here, right side here, left side here. okay? What starts to happen is you start making these little endocardial cushions. So again, those neural crest cells kind of come here, and they start making these like endocardial cushions that form from the right side here, form from the posterior portion, form from the left, and then form from the anterior portion here. So again, I have 4 total cushions. One from anterior, one posterior, one from the right, one from the left side. Then what happens is, during that process where you were kind of doing this little bit of rotation, it's starting to kind of form like these little like invagination, if you will. And what that does is it kind of like splits these little cushions here, the right and left ones, to where they kind of go like this. So look at that now. So now here you have that right endocardial cushion there. You still have that anterior endocardial cushion there, left endocardial cushion like this, and then posterior endocardial cushion like that. So again, we'll kind of give you the diagram, kind of anatomy here: anterior, posterior, right, left. Eventually what happens is, remember what we said, as you form that aortico-pulmonary septum, it rotates, right? So that corkscrew-like structure that you form, the aortico-pulmonary septum, we said it rotates. And when it rotates and kind of forms these little invaginations, it splits into 2 portions, okay? You're gonna get this posterior portion here, okay? And then you're gonna get this anterior portion here. Now here's what I want you to think about. You're gonna still have this little piece right here from that posterior endocardial cushion, right? Then a half, let's actually kind of cut this in half here with a nice little distinguishable marker here with this one. I'm gonna cut this in half and cut this in half. This portion will go here, this portion will go here, this will go here, this will go here. So now, in this little like tube, if you will, this will become where the valve is, this outflow tract. This, you'll have a posterior endocardial cushion and then half of that right endocardial cushion, and then half of that left endocardial cushion. Same thing here in the anterior portion. You'll get a half of the left endocardial cushion, you'll get a half of the right endocardial cushion, and you'll get the whole anterior endocardial cushion. This posterior structure, this posterior outflow tract, is going to be for the aorta. So this will be the left ventricular outflow tract. This one will be your right ventricular outflow tract. The left ventricular outflow tract will flow into the aorta, and the right ventricular outflow tract will allow for outflow into the pulmonary trunk. And now, what else have I formed here? My valves. These right here are my semilunar valves. And then what would these be if we think about them respectively? If this is at the left ventricular outflow tract level, that's going to be for the aorta. These are the aortic semilunar valves. If this is for the right ventricular outflow tract, which is going to feed into the pulmonary trunk, this is your pulmonary semilunar valves. And then again, not only did we have this invagination and splitting into 2 different types of outflow tracts, what else happens? Rotation. So remember how we had rotation at this point? Same thing's going to happen. We have to have rotation. And whenever we have this rotation, it puts the aorta a little bit kind of more posterior and on the right side, and the pulmonary trunk a little bit more anterior into the left side. Because you know, whenever you listen to heart sounds, when you're listening to the pulmonary valve, which side are you listening to? You're listening to the patient's what? To their left side. And if you're listening for the aortic semilunar valve, you're listening to the right side. So now, if I draw this here, kind of continuing here, I have my semilunar valves here. And again, right here. And again, this is going to be the aortic semilunar valves, and the left ventricular outflow tract. And again, this is going to be on the right side. I like to remember that because when I'm listening for heart sounds, I listen to which side? The right, like second intercostal space in an adult. Whereas over here, you're going to get your pulmonary semilunar valve, and the right ventricular outflow tract, and that's going to be on your left. Okay, I'm listening for in an adult, the left second intercostal space. So this is how I actually form these valves. So now let me just now that we've done this, what do I got to do here? Here's my outflow tracts between the pulmonary. What am I going to have right here? Pulmonary semilunar valves. What am I going to have here? Aortic semilunar valves. And then again, we've already formed them here. You can't see them, but you already have your tricuspid and your mitral valves. We have now formed the heart. All right, Ninja Nerds, in this video today, we talk about the development of the heart. I hope it made sense. I truly hope that you guys enjoyed it. And if you did like it, please continue to support us, Ninja Nerds. We can't do this without you. Ninja Nerds, we love you, we thank you, and as always, until next time. Now that we've done that, let's move to the next part and start going through what happens after we form this ostium secundum. Sounds like I'm naming transformers, right? Like the ostium secundum, all right, the septum primum. All right, never mind, let's get back to it. I gotta do that then, right?
You're like a British.
The ostium primum.
Ostium primum.
Is it the ostium primum and the ostium secundum?
Where are the English accents?
I don't know. like the Megatron or something like that. I don't know.
That was actually really good. That's good.
All right. Back down.
Can I keep that?
Yeah, you can. The ostium secundum, man. I'm gonna do it again.
Good. All right, I promise to laugh. That was pretty funny.
Good?
Okay. Yep, we're good.
All right, so now we formed the ostium secundum within the septum primum. What's the next thing that happens? All right, let's get into it, right? Sounds like I'm naming transformers right now. Ostium secundum and the septum primum, huh? All right, let's get back into it. So first thing that we have here is, what do? We formed the...
Crikey.
Dang you, Robert! Dang you!
Keep that. Crikey.
Crikey!
Where did that come from? You went straight English to freaking Dundee.
All right, we doing this again? Let's do it again.
No, we're good.
We're good? Okay.
I'm gonna keep the crikey.
Oh, frack me.